Hello, this is Harsha M, roll number 282509, and I am going to do a poster presentation on the Human Genome Project. So, the Human Genome Project was a global scientific effort whose signature goal was to generate the first sequence of the human genome. It was a large, well organized, and highly collaborative international effort that generated the first sequence of human genome and that of several additional organisms. It was carried out from 1990 to 2003, and the results obtained from this project made it a breakthrough in the field of science. Now, who carried out this project? This project did not just bring together biotechnologists, but many engineers, mathematicians, computer scientists, and statisticians. It involved researchers from 20 separate universities and research centers across the United States, UK, France, Germany, Japan, and China. And these countries were together called the International Human Genome Sequencing Consortium. Basically, when we start a project, we have to lay down the goals for it. So in case of HGP, scientists wanted to determine the entire sequence of human DNA, identify all the genes in human DNA, store this information in databases, improve tools for data analysis, transfer related technologies to the private sector, and address the ethical, legal, and social issues that may arise from this project. Now, when we look at the terms that are included in these goals, like analysis, databases, information storage, transfer of information, it is understood that biology as it was not the only subject involved in HGP. Here came bioinformatics. So, basically, bioinformatics is a field that develops software tools for understanding biological data. Bioinformatics combines computer science, statistics, maths, and engineering to analyze and access biological data. The human genome has approximately 3 to 10 to the power of 9 base pairs. If the sequences obtained from this project were to be published in a book, and if each page has at least 1000 letters, and if each book had 1000 pages, then 3300 such books would be required to store the entire genome information, which is not feasible. So, therefore, the enormous amount of data expected to be generated has made the use of high speed computer devices for data storage, its access, and analysis necessary. Hence, the Human Genome Project is closely related to bioinformatics. If you look at the methodology for HGP, there are two approaches. First, express sequence tags, which focuses on identifying all genes expressed as RNA, and second, sequence annotation. This approach involved sequencing the whole genome and later assigning functions to the different regions. Now, how is DNA sequencing done? First, total DNA is isolated from a cell and converted into random small size fragments since it is difficult to sequence long pieces of DNA. These fragments are then cloned into hosts such as bark and yak, which then amplifies each DNA fragments so that it can be sequenced easily. Next, the fragments are sequenced using automated DNA sequences. These sequences work on the principle of Sanger's method. Sanger's method, in short, is like after amplification of DNA, heat is used to denature the DNA. A primer is then annealed to the far end of the template strand of DNA. The primed DNA is then dispersed equally among 4 reaction vessels. Next, DNA polymerase is added to all 4 of the reaction vessels, followed by all 4 DNTPs into each reaction vessel. Specially modified DDNTPs, that is D-deoxy DNTPs, which lack OH at 3 carbon site, are then added to the reaction vessels. Only one type of DDNTP is added to each reaction vessel. DDNTPs basically terminate the polymerization, which can help us to obtain the overlapping fragments. So, the polymerase attaches the DNTPs to the template strand at the primer normally until a DDNTP is base paired. Once a DDNTP is base paired, the sequence is terminated because DDNTPs, as said, lacks a hydroxyl group at the 3 carbon site. As a result of chain termination, DNA fragments of different lengths are formed across all the reaction vessels. Polyacrylamide gel electrophoresis is then used to sequence the DNA. The bands in the gel is obtained and the sequence is read from bottom to top. Special computer based programs are used to arrange and align the DNA sequences based on overlapping regions present in them. Subsequently, the sequences are annotated and assigned to each chromosome. Now, this is a very time consuming process. As a result, the sequence of chromosome 1 was completed only in May 2006. If you look at the outcomes of AGP, scientists found that there are approximately 22,300 proteins coding genes in humans. We share many homologous genes with Drosophila and C. elegans, and many of our protein coding genes produce more than one protein. Our ORFs, that is open reading frames, produce 2 to 3 different proteins. A larger proportion of our genome encodes for transcription factors. They also found that there are 23 genes per million base pair on chromosome 19, 5 genes per million base pairs on chromosome 13. Humans have genes for antibodies, MHC, HLA, TCRs, and many cell signaling molecules which are not found in invertebrates. And human genome comprises of 1% of exons, that is coding region, and 99% of introns, that is non-coding regions. Applications of AGP include identification of disease-causing genes and mutations linked to different cancers. Sequence of DNA is now stored in databases in internet. AGP has helped formation of GenBank and has also benefited the advancement of forensic science and related fields. Now that marks the end of the topic. Thank you.